The structure of DNA. DNA is a double helix structure consisting of two polynucleotide strands twined around each other. Each polynucleotide strand is made up of many nucleotide molecules. Each nucleotide in DNA is composed of a 5-carbon deoxyribose sugar, a phosphate group, and a nitrogen-containing base, all linked together by covalent bonds. 4 different nitrogenous bases in DNA that can be categorized into 2 different forms, purines and pyrimidines. The purine bases are adenine, A, and guanine, G. They have 2 fused rings in their chemical structures. The pyrimidine bases are cytosine, C, and thymine, T. They have a single ring in their chemical structure. Each strand forms a backbone of alternating phosphate groups and sugars. The bases of each nucleotide are attached to each sugar and protrude inward at regular intervals along each strand. There is a constant total distance between the sugar-phosphate backbones. The 2 strands of a DNA molecule are not identical, but are instead complementary to each other. This means that a purine molecule is always paired with a pyrimidine molecule. Specifically, adenine, A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. This pairing is called complementary base pairing. As the same bases always complement each other, the base sequence of one strand can be determined from the base sequence of other strand. Hydrogen bonds link each complementary base pair. A and T are joined by 2 hydrogen bonds, while C and G with 3 hydrogen bonds. The 2 strands of a DNA molecule are antiparallel, so the sugar molecules are oriented differently. Therefore, each strand has directionality or a specific orientation. This is similar to how 2 sports teams are facing in a different direction when they shake hands before and after a game. One strand lies in 5 to 3 direction, and the other strand in 3 to 5 direction. The 5 and 3 come from the numbering of the carbons on the deoxyribose sugar. The phosphate group is on the 5-carbon, and the OH group is on the 3-carbon. Therefore, the end with phosphate group is always referred as 5-end, and the opposite end of it will be 3-end. By convention, the sequence of a DNA strand is always written in the 5 to 3 direction.